Okay, so all organisms have to develop a certain way. They have like a beginning that leads into their further development and what they turn out to be and who they are as an organism. So what happens in the first stages, earliest stages of one's development, obviously we know is fertilization, where a sperm and an egg fuse, and that kind of leads to the further development and the organism taking shape and becoming an organism. What's important to remember is that, like enzymes and other things in biology, the interaction between an egg and a sperm is very specific. They have on the sperm, it binds to the specific egg, like a specific sperm, like and an egg have a relationship. Like all of the other, there could be others, other organisms going around, but they're specifically bind to each other. So in the acromal process, that's when the acrosomes from the sperm come to the membrane of the egg, and they elicit enzymes that come in and allow for the sperm to kind of enter through the actin filaments and bind to the egg. And then once the sperm kind of enters, that's like the cortical reaction, and that's when the sperm is able to break through the layer, the vitelline layer of the egg, and then kind of move through and signal for further development. It's like enzymes, like a lock and key, so that kind of prevents the polyspermy, like all the other sperms in the environment that it could be in, from interacting and coming in. So once the sperm, lacks onto the egg, has a lock and key reaction, it kind of like zoops in and is able to come into the egg, and that like a protective layer kind of comes and protects any other sperm from entering. So once an egg is fertilized by the sperm, it's able to now undergo further processes of development. So first we have the egg, and that kind of is part of a blastomere, and that's kind of a big cluster of the different cells. And within a blastomere, there's the blastula, and that's like a single. And then within the blastula, so it's the blastomere, the blastula, and within the blastula is the blastocele, and that's kind of where everything happens. Now, that blastula and the blastomere can undergo cleavage, and that's when it breaks and it's able to prepare for further specialization. So within the blastula, there's two poles. There's the vegetal pole, and then there's the animal pole. And within the vegetal pole, there's a yolk. And through the cleavage, it's different between like the different organisms. But once the blastula cleaves, it could either be holoblastic or meroblastic. And the meroblastic is when mero is like partial, like the prefix mero is like partial, so that means the yolk doesn't really cleave, so it's kind of left as a whole, while holoblastic is when the whole cell is able to cleave. And that's kind of seen in mammals, how, I kind of think of it as like a bird's egg is different than mammals, like different than humans. Humans have the holoblastic, so that's when the yolk is able to be separated when it's cleaved and it's not kept as one, while the meroblastic, which means partial, is that the yolk is kind of left together, and like that's when you can get like chicken eggs and you can eat the yolk. So we have the blastula, and the next, it's like cleavage to the blastula, and then it kind of goes into another process called gastrulation, and that's when the blastula kind of turns into a gastrula, and that gastrula has three different parts of it. It has three different layers. It has the ectoderm, which is the outside, the mesoderm, which is the next layer, and then the endoderm, which is the final most inner layer. And within those layers, with the further development that happens, you kind of get the different types of different parts of the cell. So like the ectoderm is more of the outside of the organism. So you can get like the teeth, what's kind of more on the exterior. And while you get in the mesoderm, you can get the digestive tract, and the endoderm is when you get really in there and you into the organism, and you can get like the pituitary glands, and you know, it's kind of, you kind of have to just think of it as, if you're looking at an organism, what's on the outside, what's seen, what's like the sensory. Obviously that will be ectoderm, because you get those sensations from the outside, so it has to be exposed to the outside. Well, the innermost layer, the endoderm, that's when it's the stuff that's really in the body, like deep into the tissues, and the mesoderm is kind of all in between the respiratory, and you just have to think of it like that. So the whole process of gastrulation starts when the cells from the blastocell, like the blastosole, or so it's like the blastomere, the blastula, and within the blastula is the blastocell, with that has the cells on the outside. Those kind of detach and migrate throughout the cell. And through that, the layers that are forming invaginate, so they kind of come in, and they do so in a way that they're going to form and shape into different parts of the cell. So you're going to get the archedron, which will form into the digestive tract. And once that comes in, and it invaginates, and it'll have an opening when it comes in, and that's going to be the blastopore, and that's going to be the anus of the cell. Those are like through gastrulation, it's kind of so you're starting the specialization of the cell. So you're going to get the ectoderm, that's going to be the digestive tract, and the blastopore is going to be the anus, and that is when the mescaline cells are kind of still migrating throughout the cell. And for mammals, especially like humans, which is kind of relatable. You could kind of relate this to humans because you might be familiar with it. But the blastocyst comes, and once that's going to become an embryo eventually and form into a baby. And that's kind of the trophoblast, allows the uterine wall to kind of start to break down, and that's when the blastocyst can embed itself into the wall and form and start to develop within the uterus, and that's when the whole 9-month process begins that we're all very familiar with. Through that, there's different extra embryonic membranes that form, and that's what kind of assist the organism as it's developing. So you're going to get the amnion, which is the amniotic fluid that is part of that, which protects the developing organism that's developing. And then you're going to get the chorion, which is the site of gas exchange, and then you're going to get the allantois, and that's with the umbilical cord, and like the different nutrients come from the mother to the baby organism developing. That's where the transfer of the different blood and nutrients that it needs to develop. And then you're going to get the yolk sac, and that's the different movement through the cell, and as we were talking about the holoblastic cleavage, that's there to kind of like detect the movement. What's important to remember is that once during orgogenesis, when all of the different organs are taking shape and starting to develop, the differentiation is when the cells are migrating to the different locations throughout the cell, and that's where they kind of, once they find their place, that's where they develop, and that's what kind of determines where the different things, the organs and the parts of the cell, that's what determines what parts of the cell. Now, once they're in the locations, they're able to differentiate, and that's kind of when they specify their functions and what they're going to eventually take shape and develop into. So morphogenesis is when the cell actually begins to take shape, and then the organgenesis is when they begin to develop the different organs. And thanks to fate maps, we're able to determine the fate of the different parts of the cell and where they're going to be and what they're going to differentiate into and what they're going to turn out to be for the organism. And then cell lineage is when we're able to kind of see a cell develop through, and we can determine things from how it developed, and that is really beneficial for scientists because they're able to kind of see, and that's really eventually ultimately study the course of development in organisms. It's also important to think that development is really in the cleavage, and how when cleavage occurs, these axes form, and you're going to get the front, ventral, back, dorsal. Front, anterior, back, posterior. The anterior and posterior, that's when the organs are developed, and that's when you could kind of, the organism is matured, and that's when you're able to differentiate between the anterior and the posterior. While before you kind of developing, and it's still a cell and it's still beginning to differentiate, that's when you're going to get the ventral and the dorsal.